Hey everyone, welcome back to the Deep Dive. Today we're diving deep, really deep, into a topic that's pretty much at the heart of what you do every day as medical laboratory scientists. We're talking about those tiny but mighty cells, the thrombocytes.
Platelets.
You got it, platelets! These little guys, even though they're just fragments of cells, play a huge role in keeping us all from, well, bleeding out.
No kidding. And I know our listeners are already familiar with the basics, but today we're going beyond that.
Yeah.
We're digging into the nitty-gritty of platelet production, how they actually do their job in hemostasis, and what happens when things go wrong, like when they get destroyed too quickly.
We're talking about a full life cycle exploration here. I mean, from their birth in the bone marrow all the way to their demise. And we've got some awesome hematology articles that we've been combing through to really pull out the most clinically relevant insights, those aha moments that will make you look at a CBC or a bone marrow aspirate with a fresh perspective.
Exactly. So no more intro, let's jump right in. First things first, where do these platelets even come from? Okay, so platelet production, it's called thrombopoiesis. And this fascinating process primarily happens in the bone marrow.
The bone marrow, the body's little cell factory.
It is, and it's all thanks to these unique cells called megakaryocytes. They're the real stars of the show when it comes to making platelets.
Megakaryocytes. I always picture them as these giant cells in the bone marrow just churning out platelets.
They are big and multinucleated, which makes sense considering their job.
So how does a regular blood stem cell even become one of these specialized megakaryocyte platelet factories?
So it starts with the hematopoietic stem cell, the HSC, the mother of all blood cells.
Right, the HSCs,
Yeah.
the stem cells that can become anything in the blood.
Exactly. And for the megakaryocyte lineage, these HSCs first commit to the myeloid lineage, becoming a common myeloid progenitor, or CMP. Then they further specialize into a megakaryocyte erythroid progenitor, which is a mouthful, but basically means it can become either a megakaryocyte or a red blood cell.
Okay, so it's getting more and more specific. What happens next?
From there, it's a step-by-step progression. The MEP becomes a megakaryoblast, then a promegakaryocyte, and finally a mature platelet-producing megakaryocyte.
And somewhere along the way, the nucleus of that developing cell does something pretty unique, right?
Oh, absolutely. At the promegakaryocyte stage, the nucleus undergoes this crazy process called endomitosis.
Endomitosis. It's like mitosis gone wild.
It kind of is. A DNA replicates over and over, but the cell doesn't actually divide. So you end up with this huge cell with a massive multi-lobed nucleus.
It's like it's bulking up its genetic material to be able to produce tons of platelets.
That's a great way to put it. That polyploidy, having multiple sets of chromosomes, is essential for the megakaryocyte to be such a high-output platelet factory.
Okay, so we've got these giant megakaryocytes hanging out in the bone marrow, full of DNA and ready to make platelets. How do they actually release those platelets into the bloodstream?
It's actually pretty amazing. The megakaryocyte extends these long branching processes called proplatelets into the bone marrow sinusoids.
Sinusoids, those are the specialized blood vessels in the bone marrow, right?
Yeah, and those proplatelets, they're basically long chains of preformed platelets just waiting to break off.
So it's like the megakaryocyte is dipping its toes into the bloodstream.
Exactly, and the force of the blood flow, combined with some intrinsic breaking points along the proplatelets, causes them to fragment and release individual platelets into circulation.
It's like a controlled explosion of platelets.
You could say that. It's a very efficient way to get those platelets out there.
But there has to be some kind of control mechanism, right? Something that tells the bone marrow how many platelets to make.
There is. It's a hormone called thrombopoietin, or TPO for short.
TPO, I remember that from hematology class.
It's the big boss of platelet production. It's mainly produced by the liver and acts as a sensor for platelet levels.
A sensor. How does that work?
Okay, so TPO binds to a receptor called MPL. And get this MPL receptor is found on megakaryocyte precursors, but also on circulating platelets.
Okay, so the more platelets there are, the more TPO gets bound up and cleared from the system.
Exactly. So when the platelet count is low, there's less TPO being cleared, and that remaining TPO stimulates the bone marrow to make more megakaryocytes and ultimately more platelets.
That's like a feedback loop. Low platelets, more TPO, more platelet production.
Exactly. It's a brilliant system for keeping platelet levels nice and stable. And, this relationship, the inverse relationship between platelet count and TPO levels, it's crucial to keep in mind when you're evaluating a patient with thrombocytopenia.
It can help you figure out if the problem is with production or something else.
Exactly.
So TPO is the main player, but are there other things that can influence thrombopoiesis?
Oh, for sure. There are a bunch of other cytokines like IL-6 and IL-11 that can boost megakaryocyte development.
Cytokines, those are like the messengers of the immune system, right?
They are, and they can also play a role in hematopoiesis. And then there's stem cell factor, or SCF, which supports the survival and growth of those early hematopoietic stem cells.
It's like a whole team of factors working together to make sure we have enough platelets.
Right, and we can't forget the role of transcription factors like GATA1 and FLI1. They're essential for megakaryocyte development. And then there's the bone marrow microenvironment itself, the stromal cells, the extracellular matrix. All of that provides a supportive niche for megakaryopoiesis.
So all of this is happening in the bone marrow. And, for our listeners, this understanding of thrombopoiesis, it's so relevant to their work when they're looking at a bone marrow aspirate or biopsy.
Totally. Seeing a low number of megakaryocytes, that might suggest a problem with platelet production. But if you see a normal or even high number of megakaryocytes, but the platelet count is still low, that could point to increased destruction or maybe ineffective thrombopoiesis.
Like the bone marrow is trying to compensate, but something's going wrong.
Yeah, those morphological clues could be really helpful in figuring out what's going on.
All right, so you've got our platelets produced and released into the bloodstream. Now, what about their function? How do they actually stop bleeding?
So even though platelets are tiny and they don't have a nucleus, they're incredibly complex and dynamic cells.
They're like little first responders rushing to the scene of any injury.
Exactly.
Yeah.
And their primary function is in hemostasis, stopping bleeding. But they also play a role in thrombosis, wound healing, and even inflammation.
And a healthy person has a ton of them circulating, right?
Woo. Yeah, the normal platelet count is somewhere between 150 and 450 times 10 to the ninth per liter.
Okay, so that's a lot of platelets on patrol, ready to spring into action. What makes them so good at their job?
Well, their structure is key. Their plasma membrane is packed with glycoproteins and receptors, which let them interact with the environment and with each other. And they have this outer layer called the glycocalyx, which is important for adhesion.
So they're sticky little guys.
They are, in a very specific way. And inside, they have these different types of granules, like little storage compartments, filled with all sorts of molecules they need for their job.
Like what?
Well, there are alpha granules, which contain things like fibrinogen, von Willebrand factor, platelet-derived growth factor, and P-selectin. These are all important for coagulation, adhesion, and wound healing.
Okay, so those are the big guns. What else is in there?
Then there are dense granules, which have ADP, ATP, serotonin, and calcium. Those are important for platelet activation and vasoconstriction. And of course, they have lysosomes, too, with all those enzymes for breaking things down.
It's like a whole pharmacy inside each platelet.
It is. And then there are these internal membrane systems, the open canalicular system, or OCS, and the dense tubular system, or DTS.
Okay, those sound complicated. What do they do?
The OCS is basically a network of channels that connect the inside of the platelet to the outside. This gives them a lot of surface area, which is important for releasing those granules quickly during activation. And the DTS, it stores calcium and has enzymes that help make thromboxane A2, which is another important platelet activator.
So they've got all the equipment they need to respond to an injury. And what about their shape? I remember seeing these cool pictures of activated platelets with all these little arms reaching out.
Oh yeah, those are called pseudopodia, and that shape change is part of activation. It helps them spread out and interact with the damaged vessel wall. And they can do that thanks to their cytoskeleton, which is made of microtubules and actin filaments. This gives them structure and lets them move and contract.
Okay, so let's say a blood vessel gets injured. What's the first thing the platelets do?
The very first step is adhesion. They need to stick to the site of injury.
So they can plug the hole.
Exactly. And that happens when the subendothelium gets exposed, especially collagen. That's where von Willebrand factor comes in. It acts like a bridge, binding to collagen on one side and to the GPI-BIXV receptor on the platelet surface on the other.
So VWF is like the glue that holds the platelets to the injury site.
It's more like a grappling hook, actually, especially under high shear stress conditions, but it's essential for that initial tethering.
And are there other molecules involved in adhesion?
Yeah, there's also GPVI, another collagen receptor, and integrins like alpha-2 beta-1. These all help strengthen the attachment. And, defects in these molecules, they lead to bleeding disorders like Bernard-Soulier syndrome and von Willebrand disease.
So those are pretty serious conditions.
They are, which shows how important adhesion is.
Okay, so the platelet is stuck. What happens next?
Next comes activation. Binding to the subendothelium triggers a whole cascade of signaling events inside the platelet.
So it's like an alarm bell goes off.
Exactly, and this alarm is further amplified by ADP released from damaged cells and by thromboxane A2, which the platelet itself produces.
So what happens when the platelet gets activated?
A bunch of things. It changes shape, going from smooth and disc-like to more spiky and spherical with those pseudopodia you mentioned. It also releases the contents of its granules, a process called degranulation. And importantly, there's a conformational change in the GPIB receptor on the platelet surface, which increases its affinity for fibrinogen.
So the platelet is getting ready to clump together with other platelets.
Exactly, and that clumping, that's called aggregation, and it's the next big step in hemostasis.
And what are the key players in activation?
Well, we've already talked about GPI-XV and GPVI for collagen binding, but there are also ADP receptors like P2Y1 and P2Y12, which are really important for amplifying activation. And thromboxane A2, once it's produced, binds to its own receptors and further ramps things up. And then there's thrombin, the key enzyme in the coagulation cascade, which is a potent platelet activator too.
So lots of ways to get those platelets fired up.
Yeah, and it's important to remember that problems with granule content, like in storage pool deficiencies or gray platelet syndrome, can really mess up activation.
Okay, so the platelets are activated, they're ready to aggregate. How does that work?
Aggregation is basically the platelets sticking to each other, forming that initial plug to stop the bleeding.
And fibrinogen is the key, right?
Right. Yes, fibrinogen is like the bridge that connects the platelets. It binds to the activated GPI by IR receptors on two different platelets, linking them together.
And if there's a problem with GPI-beta, that's where you get Glanzmann thrombasthenia.
Right, a severe bleeding disorder where the platelets just can't aggregate properly.
Okay, so we've got adhesion, activation, and aggregation. What about the release reaction, that granule release we talked about earlier?
So that happens during activation. The platelet releases the contents of its granules into the surrounding environment, which is crucial for recruiting and activating even more platelets and for triggering the coagulation cascade.
So it's like a positive feedback loop.
It is. ADP and TXA2, released from the dense granules, they act as potent agonists, telling nearby platelets to activate and aggregate. Serotonin helps constrict blood vessels, reducing blood flow to the injured area, and alpha granules release clotting factors like fibrinogen and VWF to help form that stable clot. And of course, problems with granule release, like in Wiskott-Aldrich syndrome, can lead to bleeding problems too.
So we've got this platelet plug forming, but it's probably still a bit fragile at this point. How does it get stabilized?
That's where the coagulation cascade kicks in. All those clotting factors come together to generate thrombin, which converts fibrinogen into fibrin.
Fibrin, the mesh that holds the clot together.
Exactly. That fibrin mesh weaves itself around the platelet plug, binding to those activated GPII by I receptors and making the whole thing much stronger and more stable. And the platelets themselves contribute to this by contracting and pulling the edges of the wound closer together, which is called clot retraction.
So it's a team effort between the platelets and the coagulation cascade.
Definitely. And thrombin is a key player in both. It not only helps make fibrin, but it also activates platelets, creating a nice positive feedback loop.
But how does the body keep all of this in check? I mean, if platelets are so good at clotting, how come we don't just clot all the time?
That's a great question. There are several mechanisms that prevent inappropriate clotting. Endothelial cells, the cells that line our blood vessels, they release prostacyclin and nitric oxide, which inhibit platelet activation and aggregation.
So the healthy endothelium is actively preventing platelets from sticking.
Yes, exactly. And they also express ADPase, which breaks down ADP, another platelet activator. And then there's the thrombomodulin thrombin complex on endothelial cells, which activates protein C, a natural anticoagulant.
So it's like a delicate balance between pro-clotting and anti-clotting factors.
It is. And platelets themselves have inhibitory receptors too, which can dampen down activation signals.
So it's not just about activating platelets, it's about controlling them too.
Absolutely. And that balance is key to preventing unwanted clots.
Now, in the lab, how do you assess all of these different aspects of platelet function? What tests are out there?
Well, there's the basic platelet count, of course, which tells you how many platelets there are. And then there are more specialized tests like the platelet function analyzer or PFA 100, which measures how long it takes for a platelet plug to form under high shear stress.
So it simulates what happens in a blood vessel.
Exactly. And then there are platelet aggregation studies where you add different agonists to platelet-rich plasma and see how well they aggregate.
Agonists, those are the things that trigger platelet activation, right?
They are things like ADP, collagen, epinephrine, thrombin, arachidonic acid, and ristosutin. Each one targets different pathways, so it can help pinpoint where the problem is.
So it's like a diagnostic puzzle.
It is. And then there's flow cytometry, which lets you look at platelet surface markers and see if they're activated or not. And then there are viscoelastic tests like TEG and ROTEM, which give you a broader picture of how the whole clotting process is working.
So lots of tools to assess platelet function. And what about when these tests reveal a problem? What kind of disorders are we talking about?
Broadly, there are quantitative disorders, which mean there's an abnormal number of platelets, either too many or too few. Thrombocytopenia is when there are too few, and thrombocytosis is when there are too many. And then there are qualitative disorders where the platelets themselves aren't functioning properly.
Like those inherited disorders we talked about earlier.
Yeah, those are the classic examples: Bernard-Soulier, Glanzmann thrombocythemia, storage pool deficiencies. But there are also acquired disorders where something's interfering with platelet function.
Like medications or certain medical conditions.
Exactly. Things like uremia, certain drugs like aspirin and clopidogrel, myeloproliferative neoplasms, even von Willebrand disease can affect platelet function indirectly.
And all of this deep understanding of platelet function, it's so central to the work that our listeners do in the lab.
Absolutely. They're the ones who are generating the data that helps diagnose and manage these disorders.
Okay, so we've covered platelet production and function. Now, what about destruction? What happens when the platelet count drops because they're being destroyed too quickly?
So that's thrombocytopenia due to increased destruction or consumption. It's defined as a platelet count below 150 times 10 to the ninth per liter. But the key here is that it's because the platelets are being removed from circulation too quickly.
So it's not just a problem with the production, it's a problem with survival.
Exactly. And this is where it's so important to look at the bone marrow response. If the megakaryocytes are normal or even increased, that tells you the bone marrow is trying to keep up. But something's destroying the platelets faster than they can be made.
So what are the main ways that platelets get destroyed?
There are three main categories. First, there's immune-mediated destruction, where the body's own immune system attacks the platelets. This can involve antibodies binding to platelet antigens and triggering destruction by macrophages in the spleen, or it can involve complement-mediated lysis.
So it's like an autoimmune reaction against the platelets.
Right. And ITP, immune thrombocytopenic purpura, is the classic example of that.
And what are the other ways platelets can be destroyed?
The second category is non-immune-mediated destruction, where the platelets get damaged or consumed in the microvasculature. This happens in thrombotic microangiopathies, or TMAs, like TTP and HUS.
TTP and HUS, those are pretty serious conditions.
They are, and they're characterized by the formation of tiny clots in small blood vessels, which can damage organs. And then the third way platelets can be lost is through sequestration, where they get trapped in the spleen, especially if it's enlarged.
So the spleen is like a big filter for the blood.
It is, and in some conditions, it can hold on to too many platelets.
Okay, so when you suspect increased platelet destruction, what lab tests are helpful?
It's important to be thorough. You start with the CBC, which will confirm the low platelet count and give you other information about the blood cells. Then you look at the peripheral blood smear, which can show you if there are any schistocytes, those fragmented red blood cells that are a hallmark of TMAs.
So those schistocytes are a big clue that there's something mechanical going on.
They are, and you also want to do a direct antiglobulin test, or DTATE, to rule out immune-mediated red blood cell destruction. And of course, you need to check coagulation studies like PT, APTT, fibrinogen, and D-dimer to rule out DIC.
DIC, disseminated intravascular coagulation. That's a whole other can of worms.
It is, and it's important to rule it out. And depending on the suspected diagnosis, there are other specific tests like 8-MTS13 activity for TTP, Shiga toxin for HUS, and HIT antibody testing for heparin-induced thrombocytopenia. And sometimes a bone marrow aspirate and biopsy are needed to assess the megakaryocytes and rule out other bone marrow disorders.
So it's like putting together a puzzle, using all these different pieces of information to figure out what's going on.
Exactly, and that's where the expertise of a medical laboratory scientist is so crucial.
All right, so before we wrap up, can we just quickly go over some of the key terms related to increased platelet destruction?
Sure. So thrombocytopenia, that just means low platelets. ITP is immune thrombocytopenic purpura, an autoimmune disorder. TTP is thrombotic thrombocytopenic purpura, which is caused by a deficiency in the AMTS13 enzyme. HUS is hemolytic uremic syndrome, often associated with Shiga toxin. DIC is disseminated intravascular coagulation, a consumptive coagulopathy. Schistocytes are those fragmented red blood cells. Haptoglobin is a protein that binds free hemoglobin, and its levels can be low in hemolytic conditions.
That's a lot of information, but it's also important for understanding these complex disorders.
It is, and it all comes back to the role of the medical laboratory scientist in providing accurate and timely results that guide patient care.
Okay, so we've covered a lot of ground today. We started with thrombopoiesis, the production of platelets in the bone marrow, regulated by TPO. We talked about the amazing structure and function of platelets, how they adhere, activate, aggregate, and secrete to form that initial plug and stop bleeding.
Hmm.
And we ended with platelet destruction, exploring the different ways platelets can be prematurely removed from circulation.
It's been a great deep dive, and I think it highlights just how important a solid understanding of platelet biology is for medical laboratory scientists. They're the ones who are on the front lines, analyzing the data and helping clinicians make those critical decisions about patient care.
And I think it's amazing how these tiny little cell fragments play such a huge role in our health. They're far more than just sticky bits in the blood.
Absolutely. And it makes you wonder, what are some of the most challenging cases of thrombocytopenia that you, our listeners, encounter in the lab? What are those tricky situations where the diagnosis isn't so clear-cut?
That's a great question to ponder. And as our understanding of platelets continues to evolve, we're bound to find even more answers and develop even better diagnostic and therapeutic approaches.
For sure, there's always more to learn.